This lesson's about post-transcriptional modification. Eukaryotes modify the primary transcript, meaning the RNA that is first transcribed off of the DNA. We call that the primary transcript. It can also be called the precursor messenger RNA or the pre-mRNA. They modify this before it's exported from the nucleus. So this box here is meant to show the primary transcript. So this is a single-stranded RNA molecule that is represented here. And eukaryotes are going to add a 5' cap. And that's what it sounds like. It's a structure that goes on the 5' end. So if this is the 5' end, we get a 5' cap over here. And that 5' cap is made of a modified guanine nucleotide, so a modified G. And on the other end, on the 3' end, so this is the 3' end, a poly-A tail is added. So a whole bunch of adenine nucleotides, this is a poly-A tail, a whole bunch of adenine nucleotides are added, and that can be like 50 to 250 adenine nucleotides. And these give extra stability to the RNA. They help target it for export out of the nucleus, and generally they just mark it as messenger RNA as opposed to, for example, transfer RNA or ribosomal RNA. So the poly-A tail and the 5' cap, both modified versions of nucleotides, in this case one, in this case many, that go on the ends of the primary transcript. We've also seen that in the middle, the introns are spliced out. So we've seen in an earlier lesson that introns are non-coding sequences in eukaryote DNA that get cut out and never make it out of the nucleus. And so those get cut out by a complex made of RNA and protein called the spliceosome. Let's take a bit of a further look at the splicing out of introns. So here what I've drawn is, again, a primary transcript. I don't have a 5' cap or a poly-A tail here, but in the non-red colors, I've got these numbered exons. These are going to leave the nucleus. And in red, I've got introns. So the spliceosomes are going to cut these introns out, and they're going to stitch the exons together. But they can do this in a variety of different ways, and that's called alternative splicing. For example, one way I've shown here is just joining exons 1, 2, 3, and 4 together, and you get that. But perhaps exon 3 is left out, and then you get this. And remember that this is now going to leave the nucleus and go and get translated on the ribosomes. And so this has a different overall sequence, and therefore will lead to a different polypeptide with different amino acids than this one will. The amino acids that would be called for by this exon here, exon 3, are going to be missing in this one, so we'll get different proteins. And this is a way that a relatively limited number of genes can actually code for many, many more proteins because of this kind of way of rearranging and remixing the different exons. So that's a quick look at post-transcriptional modification, including adding a 5' cap, a poly-A tail, and splicing out introns. All of this happens in eukaryotes.